attending tonight's webinar. I'm Jacob Marty. I'm a financial advisor at the Verona Office of Wealth Distribution Strategies. And at WDS, our mission is to help our clients achieve their financial, family, health, charitable, and social aspirations one conversation at a time. So today we'll be having a health conversation. We'll be discussing circadian rhythm with Dr. Vanessa Leone, who is from UW-Madison. You can expect Vanessa to be talking and presenting for about 45 minutes. So during that, you can ask questions in the chat and we'll keep track of those. And then after 45 minutes over, we'll have 10 to 15 minutes that we'll be able to have some Q&A and see what Vanessa has for answers and with a short recap afterwards. So feel free to ask questions during it or even afterwards, and we could try to get you some answers. This webinar will be recorded, and a short recap version and a full version will be available to clients within about 2 to 3 weeks. So again, if you have any questions during the webinar, please let us know, and we'll be sure to ask those. So without further ado, I'll hand it over to Vanessa.
Thanks so much. I want to say thanks, first of all, to the organizers for having me tonight. This is one of my favorite subjects. I'm going to be talking about circadian rhythms and in the context of our gut microbiome. So hopefully that will be interesting to you all, and you'll have some wonderful questions at the end. So Jacob, I just want to make sure that you're seeing the correct screen with just a single slide.
Looks good to me.
All right, then we'll take off. So I titled this talk tonight, "TikTok Goes Our Microbial Clock." So thinking about how microbes actually play a role in maintaining circadian rhythms and our circadian networks in our body in the context of maintaining health. So just to give you a little bit of background about myself, I'm Vanessa. I am one of the assistant professors here at UW-Madison. I'm actually housed in one of our most beautiful newest buildings on campus, the Meat Science and Animal Biologics Discovery Building. So we sit somewhere around here on our beautiful lakeshore here at UW. And the focus of my lab is really trying to understand how our gut microbes, or the trillions of microorganisms that reside in our intestine, actually impact our own metabolism. So thinking about this in the context of metabolic health. So we think about how diet, so whether you're taking in a healthy diet or maybe an unhealthy diet, impacts the diversity of those microbes that reside in our intestines and the types of microbes that are there, and ultimately not only the types of microbes that are present in our guts, but also what can they make as part of their metabolic potential that can impact us. So what is a healthy microbiota that contributes to leanness versus perhaps an unhealthy microbiome, or at least an imbalanced microbiome that might actually contribute to the development of metabolic diseases like obesity, type 2 diabetes, and even metabolic dysfunction associated steatotic liver disease, or what we used to call fatty liver disease. So tonight, I really wanted to give you a little bit of a background and think about the role of diet and microbes in metabolism, discuss some of our initial findings that actually helped us to connect circadian rhythms of host systems, or our own systems, your systems and my systems, to gut microbes, and how we even got to this point. Then discuss some of these translational implications for some of the work that we've done in animal models and even cell culture models using individual cells, and how that might translate to human health. Think about some key takeaways, and then outline future goals for this field, what I'm going to call chronobiome field in general. So I think we can all agree that the prevalence of new age disorders has really increased over the past half century. This is just demonstrating within the United States alone how obesity has risen. This is all self-reported obesity and type 2 diabetes across our nation. So from 1985 all the way up to 2010, and 1994 to 2010 for type 2 diabetes, we can see that we've just had this progressive march for the onset of these very detrimental metabolic diseases. And we know that the situation has gotten worse. I always say when the CDC starts to recolor these maps, we're really getting into more trouble. And we can see that, where we had less than 20% reporting obesity, now we have greater than 35%. And even since COVID, which hard to believe we've already made it through a pandemic here over the past 4 years. The situation has then even gotten worse. And as of 2022, we can see that, the colors are changing even dramatically. And even Colorado now is starting to bump up with their incidence of obesity specifically. Now, I will say that with the onset of some of the newer drugs that we have on the market, all of these GLP-1 agonists that I'm sure you're starting to see commercials for on the television, Ozempic, Wegovy, Mounjaro, I'm asking the question that perhaps we're going to actually start to see a reduction in obesity prevalence across the United States. But these medications do indeed come with some interesting implications. So we have to kind of wait and see. But regardless, we can say that obesity in general has become a major health concern across our country and the world, and we really don't have many therapies that we can use to alleviate disease, aside from major lifestyle changes, which we know are really hard to implement. So with that in mind, we know that these complex metabolic diseases they're very multifactorial. So there's many factors within our environment. Certainly there's one genetic component here, but we have to think about how our genetics are interacting with all of these environmental factors that some of them we really don't even have control over. And some of them that I've really been thinking about is the connection between gut microbes, nutrition, and we can even think about overnutrition, and circadian alignment. And I left sleep off here because sleep is actually separate from our circadian system. They certainly do interact, but there's unique systems that are actually in control of sleep versus circadian alignment. So I really wanted to focus tonight on circadian rhythms specifically. But how do we even kind of think about gut microbes and their role here in driving these complex metabolic diseases? Well, we know that our diets have dramatically changed over, the past century. So where we maybe were considered more of a hunter-gatherer type to some degree, we've now shifted to taking in very ultra-processed foods. So, and probably an overabundance of some of these foods as well. So this includes foods that are high in saturated fat, high in simple sugars, and then, of course, with increased saturated fat comes increased intake of cholesterol. So what are the implications of this for our gut microbiota, who's really seeing our food first? And I think this study remains one of my favorites to demonstrate how diet is implicated in shaping gut bacterial profiles in humans. So this is more of an observational study looking at children in Burkina Faso, Africa, as compared to children in the European Union. And this is looking at the profiles of the types of bacteria that actually reside in the intestine. So this is looking at a specific gene that all bacteria express within their genome and trying to categorize them as to who they might belong to from a taxonomic perspective. And so what we can see here for the children in Burkina Faso, Africa versus the European Union, just based off of the colors in these pie charts, is that the children in Burkina Faso, Africa have a really unique microbiota composition relative to those children in the EU, where the children in the EU are dominated by what we call firmicutes, a specific phylum level of microbes, and the children in Burkina Faso, Africa actually have more Bacteroidetes. Now, when these investigators actually then look to see what component of these lifestyles of these children was really driving these changes, they noted that diet was a very dramatic, so it had a really high association with the types of microbes that are present. So here we can see that even caloric intake on a daily basis is quite different between these groups of children, and also the distribution of what we might call the macronutrients, protein, fat, and carbohydrates are really different. And I would say that more than likely the children in the EU are taking in more saturated fat and simple sugars, whereas those children in the EU are taking in less, or in Burkina Faso, Africa, are taking in more unsaturated fats and complex carbohydrates, or what we might consider fiber. So that's great that we can show association, but how about getting more into the mechanism here? And how do we really dissect the role of gut microbes in health versus disease? Because we know that many environmental factors like diet, antibiotics, even certain medications that we take in can change the microbiome. So that's great that it associates, but what about causation? And really, to understand this, I think we have to go back to our labs and our benches and try to use some of these, what we call preclinical model systems, to understand a causal role for intestinal microbes in driving disease processes. So whether that be cell culture or animal model systems, we really want to be able to ask some of these questions in these systems so that we can interrogate mechanisms and hopefully then eventually translate this to the human condition so that we can develop novel and unique therapies to intervene or even prevent. So one of these animal model systems that we really use to demonstrate causation is germ-free animals. So germ-free animals are actually completely raised in a bubble. They're completely devoid of all microbes, whether it be virus, fungi, or bacteria. They maintain long lives. They can reproduce normally. They're exposed to, completely sterile environment in food and water. And then we compare these mice to their colonized counterparts. So these have a full milieu of microbes that reside in and on the body. And what we can do is feed these animals the same diet and look to see what happens as far as metabolic outcome is concerned. So when we have mice that are fully colonized, if we feed them a high-fat diet, they actually develop obesity. However, our germ-free mouse that's devoid of all microbial communities fed the same high-fat diet actually remains lean, suggesting that microbes then are contributory to the development of metabolic diseases like obesity. What's even more interesting is that we can take microbes from our obese mouse or even human and put it into our germ-free mouse, and it can actually transfer the phenotype. So if these germ-free mice receive microbes from our obese mouse over here, this mouse will then develop obesity and other symptoms of metabolic disease. So that really suggests that there's a causative role for gut microbes in driving metabolic disruption. Now, there's lots of mechanisms that have been proposed for why Western diet or poor diet-induced gut microbiota can actually impact obesity. And so we're not going to go through all of these, but microbes can change energy extraction from the diet, so they can actually extract more energy that then our bodies can use. They can change intestinal permeability. So now you might have microbes or their products that they make translocating across the intestine into peripheral tissues like the liver, adipose tissue. Some of these metabolites might even reach the brain or impact our muscular system, which can change our insulin sensitivity and lead to obesity. It can change our gut-brain axis and even our behavioral outputs. So again, sleep-wake cycle would be an example. But when I was completing my degree, I really wanted to understand whether or not there were novel mechanisms for how microbes were playing a role in obesity. And this is where circadian rhythms come into the story. So when we were looking at our germ-free mouse as compared to those animals raised with gut microbes, we were simply asking what was different between the genes that were expressed between these two groups of mice. And what we were really surprised to see was that in nearly every tissue in the body that we looked at, there were these genes that were associated with circadian rhythm signaling. Now, lots of other genes are also being changed, but this was one that was very consistent across all tissue types. Now, we were not circadian biologists at the time by any stretch of the imagination, so we had to kind of dig into the literature to understand this. And so when we think about what circadian even means, in Latin, it means circa dies, or literally around a day, so around a 24-hour period. So that gave us an idea that perhaps microbes were actually engaging or influencing our circadian networks. And we wanted to dig into this and understand this a little bit more because we thought that could be really important for metabolism. And why is that? Well, it turns out that circadian clocks are highly involved in regulating host physiology, particularly over a 24-hour period. So while these clocks are endogenous within different tissues within the body, nearly every tissue expresses these genes. Really, our central clock located within our brain is the one that's in charge of detecting what we call photic cues. So this would be things like light-dark cycle, or when the sun comes up or goes down, or even, as we know in our lifestyles now, sensing artificial light. So this actually then communicates through the retina to a specific region in the brain called a suprachiasmatic nucleus within the hypothalamus. And this is actually in charge of regulating all or many of the peripheral clocks in our body. So those located in the liver versus our fat cells, even within our intestine. And this really guides our bodies into understanding when we should be performing something like physical activity versus sleeping, which then can even feed into timing of feeding and the types of nutrients that we're taking in. Now, we think gut microbes are really important for this. And when we think about these photic cues or these signals that are actually driving our circadian clocks, we oftentimes use this word Zeitgeber, which literally German means light giver. So that would be a cue for our circadian networks to become activated and elicit this rhythmic signal when we have light versus dark cues, as an example. I think this system is really interesting because while the suprachiasmatic nucleus is really in charge and it's what we call our master or central clock, all of our peripheral clocks can also be sensitive to external cues. So if we're eating at different times of day, our liver might respond differently and start to pull away from our master central clock and start to run on its own time. But when things are healthy and running in the correct way, most and nearly all of our clocks are aligned and exhibiting the same day versus night or rhythmic pattern. Now, we know that there's many situations that can be involved here for disrupting the clock in particular. So we can think about how we might be exposed to artificial light. There could be genetic mutations. So believe it or not, there's certain genes within the core circadian clock network that when they become disrupted, they can then change the rhythmic patterns of that individual's day versus night outputs. We can have social jet lag. So for those of you who like to sleep in on the weekend and stay up late, that can be a disruptor for your circadian networks. People who experience shift work or sleep apnea or truly undergo jet lag where they're traveling, to China and back within a short period of time. And even simply by changing our feeding pattern, so late night feeding or even shifting to a high fat diet. But with our finding that germ-free mice versus those that were raised with gut microbes have unique circadian clocks, we really wanted to understand how microbes were a central player here in driving circadian networks. So we went back to the drawing board, our drawing board of mice, really, and we were comparing our conventionally raised animals to those mice that were germ-free. And we fed them, again, these same diets. And as the literature had told us, those mice that have gut microbes, when we feed them a high-fat diet, their clocks become broken or disrupted, which can then alter metabolic outputs, disrupt them really, and lead to obesity. And so this means that, compared to our normal low-fat, high-fiber diet-fed animals who remain lean, our high-fat diet-fed animals really exhibited this clock disruption. Now, we wanted to know then, what's going to happen here in the germ-free case? Is diet enough to disrupt the germ-free mouse circadian clock, or will it look the same? And to our surprise, it turned out that regardless of the diet that the germ-free mice were fed, the circadian clock certainly looked different. It was not broken as we would have anticipated based off of our colonized animals, but the clock was running different regardless of the diet they were fed, and these animals remained lean. So we really thought, well, what is the microbiome making that the host is then seeing that's contributing then to this broken clock in this case, and that might be absent here that's causing these animals to remain lean and run on a different time scale. So when we actually then examined the gut microbiota, what we really observed was that our gut microbes are actually there to sense dietary cues, which translates into these outputs that help to maintain circadian networks, both in the central clock in the brain as well as in peripheral clocks. So that means that gut microbes are actually there to sense when we're eating, how much we're eating, and really what are we eating? So what does our diet composition look like? And this translated into day versus night patterns of just what the microbes were able to do with their metabolic potential. So when we were eating a diet like this, we had these nice day versus night patterns as to what the microbes were actually able to do. This translated into what we call a microbial metabolome, where all of these signals then can be absorbed into host circulation. And what we saw was that both central and peripheral clocks were also aligned and exhibiting beautiful day versus night patterns. Now, and this ultimately resulted in wellness, at least metabolic wellness in these animals. Now, if we hit the system with the high-fat diet, we saw something very different, where we were now changing the way these mice were actually eating. So they're eating more during the day. Mice are nocturnal, so they should really only be eating during the dark. So they were eating more during the day. This was causing a dampening of microbial rhythms. It was disrupting the microbial metabolome. And now the central versus peripheral clocks were seeing metabolites at perhaps the wrong time of day, which was leading to then disruption of these core circadian clock networks. And then that can contribute to the development of obesity. And I want to point out that this is definitely a bidirectional communication that's going on. So if we disrupt either by genetics, which we can do in mice, and then there are some human genes that we know can be disrupted, or we do something like a jet lag model in mice or in humans, we can disrupt these networks, these core circadian clock networks in different tissues. And that can feed back onto our intestinal clock, disrupt microbial oscillations and the metabolome, and contribute to the development of obesity. So, this is a really important bidirectional communication that's going on between microbes and us as the host to understand what's going on within the environment to drive these systems to stay, if you will, in time. So we next wanted to ask, what are these microbially derived metabolites that are really important in the context of circadian rhythm? So how are these metabolites and what are these metabolites that are contributing to maintaining these networks? And when we consider what this healthy diet looks like, low fat, high fiber diet, we know that there's fermentable carbohydrates, fibers, that really only bacteria can metabolize, and they can produce what we call short chain fatty acids. So these are 3 of the most common short chain fatty acids produced by gut microbes from fiber fermentation: acetate, butyrate, and propionate. And it turns out that short chain fatty acids actually account for 10% of the calories of any given human of their caloric intake in per day. So that means that microbes themselves, based off of their own fermentation patterns, contribute to our caloric needs. And it turns out that these cells that line our intestine, the epithelial cells, use these short chain fatty acids as their primary energy source. So it's really important that we actually take in fiber and have the right microbes there to ferment it to produce these short chain fatty acids. And then there's a number of other metabolites that can be made through these bacterial fermentation processes, specifically tryptophan metabolites. And so many of you have heard of serotonin and melatonin, and tryptophan sort of falls into that category of a metabolite that's important for serotonin production in the body. And these can all then elicit health effects. So we were really interested in these short chain fatty acids in particular, given their beneficial health effects for the host. So what is going on with butyrate over a 24-hour period? So if we just look at our mice that are colonized with bacteria, our germ-free mice, because they don't have any bugs to do fermentation, shouldn't have any butyrate in particular. And so when we think about our colonized animals that have gut microbes and are fed a healthy diet versus an unhealthy diet, we can see that our healthy mice fed this, what we call a regular chow diet, have a beautiful diurnal pattern. So this is a 12-12 light dark period. So this is showing a beautiful day versus night pattern in the levels of butyrate that are found within the intestine. But when we feed this poor diet, we see that we lose that rhythm almost completely. So that's telling us that diet is really important for driving the types of microbes that are there, and then the microbial metabolites like butyrate that they can make over a 24-hour period. And when we even then take our germ-free mouse, who should not have butyrate because they don't have any bugs to make it, and now we expose that germ-free mouse to butyrate just by simply injecting it with butyrate, as compared to what we would call a control, we can see that the time of day that we actually administer the butyrate has a profound effect on a peripheral clock like the liver. So this is what the clock expression of two key circadian genes would look like if we look at the ratio of their expression. And we can see that if we expose animals to butyrate in the dark cycle, when they should be active, we can actually shift that ratio of gene expression. And this is really only happening in the liver. It's not happening in the core central clock of the body. So butyrate specifically seems to be able to sort of help to reset our peripheral circadian clock gene expression, which is really interesting. And the implications of this for metabolism are still really being investigated in my lab and in others. So, I think it's really great that we can have these findings in animals. But again, I said we like to translate our findings to the human condition because that's the most important thing. Or we can even think about our companion animals as well, who also experience obesity and other metabolic diseases. So there's lots of differences between mice and humans. This is what a mouse eats, and this is what a human eats, right? So the diversity of the diet is quite different. And as I mentioned, mice are nocturnal, whereas humans are diurnal. So we are most active during the day and we sleep during the night. So how did these findings then translate? So I had the fortune of being able to collaborate with a group that actually does controlled in-lab experiments with human subjects. So in this case, we actually had 9 healthy individuals. So they had normal BMI that were maintained under a normal or a sleep restricted schedule. And this was a crossover design. So both people went into both arms, the subjects went into both arms of this study. And what we were able to do was actually keep them in the lab, in the clinical lab, for several nights, and they were allowed to habituate for a night, and then they were allowed to have a normal sleep schedule, or what we did was actually shorten their sleep schedule by like 2 hours on either end. So this was what we call a restricted sleep schedule. What was really interesting about this was that the meal timing was maintained and the meal composition was maintained to be the same during both arms of these studies. And under these conditions, we were able to perform 24-hour blood sampling. We weren't able to collect stool from these individuals. As you can imagine, it is kind of difficult to collect stool from a person over a 24-hour period. So we were relying then on blood. So with that, we could process these blood samples, we could isolate the metabolites. We could use some fancy serum metabolomics analyses, what we call untargeted metabolomics. We can get features of these metabolites that are found within these blood samples. And then using a bunch of reference databases and quality control metrics, what we were able to do was whittle this down to 90 features or 90 metabolites, and then categorize them as being either microbially derived, those that were derived either from microbe or host, because we can make some of the same metabolites, derived from host metabolism of a microbial metabolite, or only those that were diet or host derived. That was how we were able to categorize them. Then we could perform some fancy analyses to understand which metabolic pathways these metabolites fell into, and whether or not they actually exhibited rhythmic patterns like I just showed you previously in our animal model system. So as you can imagine, 90 metabolites, we had diverse categories of metabolites that were detected in serum. And some of these indeed fell into those that could be microbially derived. So we did detect short chain fatty acids, specifically butyrate, some tryptophan metabolites, again, that could be made by gut microbes, as well as some other things like bile acids, fatty acids, and even amino acids, which we know are so important for host physiology. So what we were really interested to see is that when we had this restricted sleep schedule, and we think about this red line that I'm showing here, we actually saw a dampening of the rhythm following sleep deprivation or sleep restriction. And when we applied statistical analyses, we saw that the oscillation or the diurnal pattern here was actually absent following sleep restriction. So you can see that this peak here in particular was lost following sleep deprivation. And again, these are the same individuals that were tracked under both conditions. So that told us that perhaps some of what we were seeing in our mice by feeding them a high-fat diet could be recapitulated in humans who are only experiencing sleep disruption. So again, another way to kind of target that host circadian system. And next, we looked at some tryptophan metabolites. So again, many of you are familiar with serotonin, and maybe we call that the happy gut hormone, which is derived from tryptophan specifically. And tryptophan, the bugs and our own bodies can do lots of really unique and special things with tryptophan. So our own bodies can make serotonin, and that can be converted into N-methylserotonin. Both microbes and the host can make kynurenine. And then microbes can do some really cool things. They can make some indole compounds, indole acetate, indole acetaldehyde, and most importantly, indole 3-propionic acid. So IPA has been shown to have and correlate with actually host health. Where something like kynurenine has actually been shown to be elevated in people who have anxiety and depression. So we're really interested to see what the patterns look like before and after sleep deprivation, because we know what can happen when we're sleep deprived. So as we expected, the main parent compound here, tryptophan, exhibited diurnal patterns regardless of the sleep intervention. So we were not impacting oscillations or day versus night patterns of tryptophan here. But when we actually started to look at some of these other compounds, like N-methylserotonin, which our own bodies make, or indoxyl sulfate, which our bodies make from indole, which is made by gut microbes, the patterns didn't change here either. So that was interesting to us. But kynurenine and IPA actually showed a very different pattern in that under baseline normal sleep conditions, kynurenine actually did not exhibit a diurnal pattern. It was only gaining an oscillation under restricted sleep conditions. Now, IPA was different because under normal sleep conditions, it was exhibiting a pattern that became disrupted following sleep restriction. So while this is still very observational, we find this to be really intriguing that us, simply shortening the sleep time and doing some sleep restriction on either side of the center of our sleep pattern can elicit these changes in microbially derived metabolites that are actually making it to the periphery. The implications for this, as I said, are still unclear for the mechanistic connection here. But what we are able to say is that we can measure these metabolites over a 24-hour period in human serum, and that includes those that are microbially derived. We can detect oscillations in these metabolites in human serum. Some of them exhibit significant oscillations or day versus night patterns, and then some actually change depending on sleep status. And, perhaps some of the observed alterations in the metabolites could then contribute mechanistically to the deleterious effects of sleep restriction. So, this could be metabolites that would be involved either in metabolism or cognition, depression, anxiety. And that really ties into what we saw with kynurenine and IPA and the differences that were observed following sleep restriction. So I think in order to really understand this, we have to go back to our animal model systems to see if we can really interrogate and dig deeper into what the implications are for a change in the day versus night pattern of these specific metabolites on metabolism in the host to understand how they might contribute to something like obesity or type 2 diabetes. So as I said, we really want to understand how do microbes then maybe contribute to restoring circadian networks? So do they actually play a role in this restoration? And this is not work of my own. This is work of actually one of my very good colleagues, Amir Zaranpar. What he showed was that if you time restrict feed even a high-fat diet, and this is what it was done in mice, what you can do is restore some microbial oscillations, which then restores some of the microbial metabolome, which can then aid in realigning circadian networks and contribute to the prevention of the development of obesity. So not all is lost. We can use things like time-restricted feeding to actually restore some of these oscillations. And I just want to show, some data that's been done in humans to show how we can use early versus late eating and actually examine this from an experimental perspective to see how it impacts microbes. So as I mentioned, it's very difficult to collect stool from any individual over a 24-hour period. It's really easy in mice. They poop a lot, as I'm sure you know if you've ever had them in your house. But we can't do that in humans. So some of the studies that have been published are actually relying on the microbiota that is actually found in the mouth. So this is actually looking at the salivary microbiota and looking at how early versus late eating in healthy humans can actually impact the patterns that we see in the salivary microbiota. So in this case, they used 10 healthy women, and this was a crossover design where they had either early versus late eating, and they sampled a certain region within the mouth specifically. So these individuals had normal BMIs. And so they actually sampled the salivary microbiota at 4 time points over a 24-hour period to look to see for those community members that are there in the mouth, was eating and time of eating important for driving the presence of certain microbes. And I won't go too far into the weeds on the data, but what they found was that there were unique diurnal patterns in microbes within the mouth that were observed based off of whether or not the individuals were having early versus late eating patterns. And if they looked at these individuals before or after early versus late, they saw that there was an inverted pattern in microbial diversity, so the types of microbes that were there that were observed when they were shifted to late eating. And eating late actually increased certain taxa that have been, again, associated with inflammation. So we, that old wives' tale of timing of feeding and when you should, have your last meal, there might be something to that, and our microbes might actually be involved in that too, even those that are found within the mouth. Now, this is a study that I would really like to participate in. They didn't ask me, so I wasn't able to participate in this study, but they were actually looking at how time of chocolate intake actually influences gut microbes and other parameters related both to sleep and metabolic health. So in this case, they had 19 postmenopausal Caucasian females that were approximately 52 years of age with normal BMIs, and they used a randomized controlled crossover trial. So individuals were exposed to either taking in evening chocolate or morning chocolate. And we're not talking about just a little piece of chocolate. This is like 100 grams, almost a full bar of milk chocolate. And so the individuals underwent this for 14 days. And then they crossed over either from evening chocolate to morning chocolate with a washout period in between. So it's interesting because chocolate does have kind of a bad rap, especially for weight gain, because it is a high energy, high sugar food. So these researchers wanted to ask if time of feeding could impact chocolate's effect both on metabolism and what was going on with the gut microbiota as well. And so they made some very interesting findings, I think. So again, they had a no chocolate, evening chocolate, or morning chocolate group. And what they showed, again, with short chain fatty acids, was that when we think about evening chocolate in particular, it appeared to increase slightly the levels of acetate, one specific short chain fatty acid, but evening chocolate definitely increased the levels of propionate when chocolate was taken at night relative to both the morning and no chocolate intake at all. Now, these researchers detected very low levels of butyrate, so I'm not able to say whether or not the day versus night patterns of butyrate production were altered by evening versus morning chocolate, but we can infer maybe some things from these other short chain fatty acids that were made. So this is a way to kind of stratify the microbiota and get a visual look at how different these microbial communities might be. So this is called a principal coordinate plot. All of these individual abbreviations here represent an individual person. And how close they are means that the microbial community is more similar. So no chocolate microbial communities are very similar. Morning chocolate, those communities are looking relatively similar versus evening chocolate, which again look relatively similar. And when the individuals were taking in chocolate just in general, their communities were shifting away from taking no chocolate at all. So this is telling us that the intake of chocolate alone is shifting the microbial community membership, which is likely contributing to these findings in these outputs here that were measured. But evening versus morning chocolate is also causing the stratification of the microbe populations that are present. And this kind of popped up with some specific microbes of interest that are involved in making things like short chain fatty acids. So this is looking at the percentage of these microbial community members here. These are specific microbes as compared to the short chain fatty acid acetate. So The ruminococcus percentage is increasing when chocolate is present, whether it's morning or evening, relative to no chocolate at all. And that's correlating with increases in acetate. And sort of the inverse is occurring here with this lactospiraceae community member, where it's increased when we have chocolate along with acetate, and then there's a decrease here. Or an increase in the lactospiraceae community member and a decrease in acetate when we don't have any chocolate intake at all. So the findings were really interesting when we think about metabolism here too, in the context of this large intake of chocolate. So there were no overt changes in weight, believe it or not, between morning chocolate or evening chocolate, but the participants, regardless of whether or not they were eating it in the morning or in the evening, spontaneously reduced their total caloric intake. Cortisol levels were actually decreased when individuals were taking in morning chocolate, and morning chocolate enhanced lipid oxidation or fat burning potential and actually improved fasting blood glucose levels. So that would be maybe an indicator that something associated with, type 2 diabetes was actually improved here if taking in chocolate in the morning. And then evening chocolate actually increased skin heat dissipation and promoted regular sleep episodes. So, I think from a metabolic perspective, morning chocolate was having better improvement on metabolism than evening chocolate, but evening chocolate was perhaps altering our sleep schedules. So I thought that study was very interesting to kind of connect this idea of how diet, timing of food intake, and microbes might contribute and interact with one another to metabolic health. So the one last point I wanted to make about the types of microbes and the day versus night patterns is their capacity for predicting the development of metabolic diseases. And so this was an extremely interesting paper that came out a couple years ago, where the researchers had access to data from a regionally similar, very large cohort of individuals. So this cohort included healthy people with obesity, type 2 diabetes, cardiovascular disease, and cancer. And so they were looking at the microbial signatures here in the stool, and they also had all of this data around host parameters like BMI, glucose levels, dietary intake. And what they realized was that they actually had time-stamped samples. So they knew whether or not the samples were collected in the morning, middle of the day, in the night, and the time of night that they were collected. And then they were looking at the microbial community membership here. And what they noted was that they could actually stratify healthy versus type 2 diabetes individuals based off of the rhythmic taxonomic features, so day versus night patterns of the types of microbes that were present in healthy versus type 2 diabetes. Turns out that type 2 diabetes actually had lost rhythms in many of these microbial features, and that separated them from healthies who had beautiful day versus night patterns of these types of microbes that were present. So with this type of information in such a large, rich data set, we can do something that we call unsupervised machine learning. And Jacob's probably very familiar with this because this is how you probably make some decisions on what you're going to, put some of your resources into. But as far as the microbiome is concerned, this allows us to identify what we might consider to be arrhythmic taxonomic risk signature within these people. So they identified 13 community members of the microbiota that were very different between the type 2 diabetes and healthy individuals. And then they kind of test this algorithm to see, can they stratify healthy versus those with high risk for type 2 diabetes just based off of these 13 microbial community members. And these were very good at predicting who was healthy versus who was and had type 2 diabetes. And then they also, because they had these data collected over time, were able to actually predict some of those individuals who maybe were pre-type 2 diabetic, that they were actually then going to develop type 2 diabetes, just simply off of these 13 microbial community members. So this is kind of like next level utility of the microbiome, using it as a biomarker really of health. And also the signature, the day versus night patterns that are present in the microbiota, and how that can be kind of leveraged then to predict healthy versus those that might have metabolic disease like type 2 diabetes and maybe even use that to examine and predict that trajectory of disease. Because then we might have the chance to intervene with either timed delivery of certain fibers or probiotics, and things like that that could restore that microbial community. So I think some key takeaways are that time of feeding coupled with diet composition are really primary drivers of that microbial community membership and the microbiome's day versus night pattern. So diets rich in diverse fiber sources that might promote the abundance of short chain fatty acids, as an example, can influence our own circadian networks. So those metabolites can actually be absorbed by the host and then elicit an influence on our circadian rhythms. We saw that short-term sleep disruption, even when we held meal composition and timing constant, was eliciting shifts in day versus night patterns of microbial metabolites in circulation. And we really need to understand then what these implications are for host metabolism. And I just want to say this to all of the attendees on the call. If you're ever asked to participate in a study examining the microbiome, remember to timestamp your stool sample, because it's really important for the researchers to be able to go back to that stool sample and understand when it was collected, because then we can use it for things like trying to do predictive modeling and things like that. And also, it really helps us to understand why we see so much heterogeneity in the microbiome across individuals if one person is providing a sample from the morning and the other is providing a sample from the night. So from a futuristic perspective on chronobiome, I think the gut microbiome is really a key feature of maintaining circadian networks and metabolism. So we can't deny that light and other factors, temperature, things like that are really important, but the trillions of microbes that live within our intestine are really important for helping to maintain these networks. Time delivery of prebiotics or probiotics, or what we might even call postbiotics, like butyrate would be considered a postbiotic. Maybe those could actually impact and influence host health if we can figure out when we should be delivering them to any given individual. I think understanding key aspects of health, so thinking about sleep-wake patterns, feeding patterns, dietary intake, work schedules is really essential. So these diurnal patterns of microbes could even be leveraged to predict development of metabolic diseases in the future, which would be really impacting, I think, as from a societal perspective. And also, I want to also really get the point across that there's really no one size fits all approach. So I'm sure there's many owls versus larks in attendance tonight, and it's really understanding that everybody has a very unique chronotype, and that should really be considered when we're making decisions about our health and how we might be able to improve it. And thinking about how the microbes can actually fit into this is really essential. So there's lots of people I would like to acknowledge. So for some of the work that we've done, both when I was a postdoc at University of Chicago and now that I'm here at University of Wisconsin, in particular, I want to highlight Evan Chrysler, who's really taken on a lot of our circadian work that we're doing here at UW-Madison that I'd love to talk more about in the future if the opportunity presents itself. And of course, all of the funding that we've had to help us make some of these really interesting insights. So with that, I want to say thank you, of course, for listening and taking time out of your evening. I know everybody's so busy, and I'm happy to take any questions if anything has really tripped your trigger.
Well, thanks, Vanessa. Lots of information in there. I was coming up with a bunch of questions, but yeah, if anyone has any questions, feel free to write them down in the chat and I'll relay them to Vanessa. So I guess just recently you were talking about the chronotype. Could you go into more detail about what that is, or like what different types there are and how you could find out which one you are?
Oh, sure. Yeah. So, well, I think we kind of all have a feeling of whether or not we're a morning versus evening person, right? And that kind of fits into this idea of owl versus lark. So even if you go to participate in a sleep study or go to see your doctor and endocrinologist to understand more about what type, what chronotype you are, they will likely give you a survey that helps to distinguish whether or not you're an early morning person versus a late night person. And I think understanding that is part of the chronotype. And that's not just like sleep-wake pattern, it's also underlying metabolic outputs like glucose tolerance, levels of cortisol, day versus night patterns, and things like that. And that kind of all fits into what we consider to be our chronotype as an individual. So from a just slang perspective, we would call it owl versus lark.
Gotcha. Thank you. How you did all these studies that are focusing on food and has a lot of questions like breaking off from that. Has there been anything about how alcohol might disrupt circadian rhythm?
Yes, yeah, there's actually a group at Rush University in Chicago that has done a lot of work looking at alcohol intake and how it disrupts circadian rhythms. And they were actually using nurses in the hospital who undergo shift work in particular. And so higher alcohol consumption, particularly in those undergoing shift work, can be really detrimental for health and associate with many downstream negative health effects like atherosclerosis, cancer, a lot of disruptions in normal metabolic processes are affected by alcohol consumption and timing of alcohol consumption. So, there is some discussion about even those who might be suffering from alcohol use disorder, if they can somehow shift their intake to a little bit earlier in the day, that can elicit an improvement in certain negative implications for the liver function in particular. So yeah, alcohol is a big one.
Okay. Thank you. There was a question that came in before from someone that worked night shift for a long time. You mentioned that a little bit there with like nurses. And they asked, what can they do at this point to fix it? They had those work scheduled for over 30 years. Are they out of luck, or is there something that they can do to fix it?
Yeah, no, I found that question to be really interesting. I kind of pondered it a little bit all day. Well, the good news is that if that's been a consistent shift, like those who work third shift as an example consistently, that's actually better than those who are shifting back and forth, right? Because there's some consistency there. So even if you are a lark and now you are working the owl shift, but if you're doing that consistently and you're maintaining maybe things like sleep hygiene, right? Like when you're actually going to bed versus when you're eating and you're really thinking about that and trying to maintain that, that's actually better than those who have to shift back and forth constantly. I know that person said like I'm going to be doing this for the next 5 years and then what? um, and there is a lot of research out there to suggest that you can shift yourself back to a normal schedule, like most of us would experience, but it takes time. So it's not something that you're going to be able to do just within a week, right? Like they oftentimes recommend shifting your sleep schedule by like 30 minutes a day in order to completely like invert your sleep cycle. So those are things that, could be considered when the time comes to do that. We know natural light is really important for sort of turning on our circadian rhythms and helping to maintain the cyclical nature of them. And then I think timing of feeding and eating is really important. So for those who actually travel and undergo jet lag, many of my colleagues who are in this field discuss and describe their fasting when they're actually traveling. So rather than our tendency to grab food off the shelf and just sort of eat whatever we want because we're tired and, our bodies are kind of telling us to do that, they actually avoid that completely. And they try to reduce their caffeine intake during their traveling bouts and also undergo a fasting cycle as well to sort of stay in alignment then even when they're traveling. So yeah, those are some tips maybe. Maybe that might be helpful.
Okay. There's another question. I'm just gonna share my screen here, which has our next client events. Just give me a second here while we ask a few more questions. Do one more.
Yeah, and I do see that there is a question about race and ethnicity in the microbiome, and I think that's a really important question. There is work to show that there are dramatic differences between people of different races and ethnic backgrounds. Um, some of that is indeed driven by genetic parameters that are obviously different, but there's also a lot of environmental factors that are quite different as well. So diet, right? And different types of diets that are taken in across different ethnicities as an example. So yeah, that does have a really big effect. From a circadian perspective, that I have more difficulty answering because I'm just not as familiar with the work that's been done from that regard. But I will say that our human study that we did, we had a nice distribution even across the 9 individuals of different races and ethnicities as well.
Okay. You were talking a little bit about prebiotics, and they're obviously marketed quite a bit. Is there stuff that you should be looking for on when you're picking them? Do they work at all? If you're trying to regulate your circadian rhythm, which ones should you look for? When should you take them during the day? That type of thing.
Yeah, so I'm actually a bigger believer in prebiotics than even probiotics. So probiotics would actually be the microbes themselves that you would buy off the shelf. So you can go to Whole Foods and look at the vast array of probiotics that they have available. But prebiotics would be like targeted fibers like fructooligosaccharides, inulin, other over-the-counter fiber sources that we might take in. And I it's difficult to say because everyone's microbiome is so different. So like the fibers that my microbiome can actually ferment might be very different from yours. So I think you kind of have to see first what you can tolerate, right? Because some individuals do have a really hard time tolerating certain fibers. So I would say that that's an important aspect because if you can't tolerate it, you're not going to keep taking it. But then also to not just think about purchasing prebiotics, but also thinking about whole foods, right? And there's a lot of research out there right now thinking about fermented food. So for those from Wisconsin, sauerkraut is not a big deal, right? Lots of us eat that. And I would say that there's probably lots of good things from that fermented product that the microbes have already made, more from like a postbiotic perspective, as well as the microbes themselves that are contained within those products. And thinking more along the lines of like that, how you can use a whole food actually to help bolster your microbiota.
Are there any cooking oils that I know they talk a lot about where you try canola oil, coconut oil, olive oil that have an effect on the microbiome? I've heard, I think coconut oil is really good for butyrate, if that's correct, or
Yeah, that's a great question. We actually did a study comparing things like safflower oil versus a milk fat based fat. And there's actually a lot to that. So the Mediterranean diet, which is still promoted as being one of the most beneficial, I think, for metabolic health, that promotes a lot of olive oil as an example. Lots of those lipids that are in there, like the medium chain fatty acids that you're referring to that would be in coconut oil, definitely can have a beneficial effect on the microbiota, but then also on the host too. So that crosstalk is just improved between host and microbes. So yeah, I think lipids are really important to consider, and we need to do more work to understand that because we know that even certain microbes can make and convert certain fats to beneficial fatty acids, like those who might be familiar with conjugated linoleic acid, that's actually can be converted by microbes from linoleic acid, and that can have lots of beneficial health effects as well. So yeah, that's a really good question and a good point.
um, do additives and preservatives, you were talking about the high fat diets and those examples were fast food and processed. Do high fat foods that don't have additives and preservatives behave differently, or are they pretty similar?
Oh, that's a good question. Yeah. Yeah. So from a research perspective, our mouse diets, they don't have too many preservatives in them, but yes, human diets have tons of preservatives in them. And there is some research to suggest that these can have negative effects on the microbiota, which then can make certain diseases worse. So like inflammatory bowel disease is an example. So there's been some research to show that certain food additives and preservatives change the microbiota, which then can have negative implications for inflammation in the intestine in particular. So I think those are really important things that should be considered that we're learning more about. And even some of the work that we're doing in our lab is using things like high fructose corn syrup, which isn't a preservative, but it's definitely a major component of ultra processed foods, and how that can have implications for the microbiota, which can make things like fatty liver disease worse, as an example. So yeah, I think there's a lot to be learned from there and understood for sure.
All right. Well, it's 7 o'clock, so as much as I would like to just keep asking lots of questions, I'll cut it off there. And if anyone again has any other questions that arise, or if you're watching a recording, send us an email and we can relay it to Dr. Leon and get it back an answer to you. Just wanted to preview, we have our next couple of events that are coming up in October, November, and December. So October will be our pumpkin carving and decorating contest. So there are prizes, fun way to just celebrate the fall spirit. November, we are going to be doing a charity drive. So all these events have more information on our website, and we'll be supporting very worthwhile charities in the area. And then in December, we're going to be resuming our Next Generation series with helping younger children, teenagers, and young adults with their financial future. So I want to thank you so much, Dr. Leon, for sharing your work and giving us some perspective on this health topic that I think is becoming more and more interesting and getting a lot more attention on the effect that it can have as we are dealing with more health consequences and in general our population dealing with this, and what type of solutions that we can apply to our personal life. So again, thank you so much for sharing all your knowledge and expertise, and thank you everyone for participating, and we'll see you hopefully participate in October and see you at the next one in December.
Yeah, thank you again so much for having me. It was really nice, and for all of the questions, and feel free to reach out. I think my email's on the slides, so I'm happy to field any questions. So thank you.
Yep, they'll be posted on the recording later too. So, well, have a good night, everyone, and thanks again.